cool.
As you can see, a cell has many organelles. They all have a specific and important job. It would take a very long time to go over all of them, so for this video I will focus on the ribosomes and the nucleus and explain their function and what chemistry is involved with them. Ribosomes are involved in protein synthesis. I will explain a basic version of the mechanism of the ribosome's job. A code for a protein is sent from the nucleus to a ribosome. The ribosome then processes the code and begins creating a chain of amino acids. The protein's shape will be determined by the order in which the amino acids are converged. This video will give you a visual on the process, and by the way, it sounds like it's voiced by a female robot.
There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life.
Probably the most important organelle in a cell is the nucleus. The nucleus isolates its contents from the cytoplasm. It often sends signals to the rest of the cell. These signals usually make the receiving organelle execute its job. Or, like I mentioned earlier, it can tell the ribosome what protein it should produce. It contains DNA, the genetic coding of a cell. DNA contains all the information needed for a cell to survive. DNA has many distinct chemical properties. Most people, probably excluding Sineath, know that its shape is a double helix. The reason for this is the intermolecular forces between the molecules that make up DNA. Each DNA segment has a phosphate group and one of the 4 nucleotides shown here. The order of the nucleotides is what determines the way the information is processed. The nucleotides are held together with hydrogen bonding. The helix forms because of the London dispersion forces between the molecules on each side. DNA clumps up very tightly. This allows a lot of DNA to remain in a small spot. They wrap up to make the shape of chromosomes. Chromosomes should be used in advertising, as shown in this picture. But how do you tell the sex of a chromosome? Just pull down its genes.
Britt, get back over here.
Here's a video about DNA, and good news, Lady Robot is back.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells.
So Cartman, how do you feel that there is only one topic left in this awesome video? And how about you, Steve Carell?
No, God! No, God, please, no! 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 Give me fuel, give me fire, give me that which I desire!
Cells obtain energy by processing food molecules that enter the cell. Complex organic food molecules such as sugars, fats, and proteins are rich sources of energy for cells. Much of the energy used to form these molecules is stored within the chemical bonds that hold them together. Cells receive the energy through a series of oxidation reactions with the food molecules. Once a cell harvests the energy, it stores it as ATP. So when Miss Stripmodder takes your food, don't take it personally. She's simply trying to store energy for her cells. The most well-known energy process is called glycolysis. A cell breaks down glucose through many complex steps. Each step requires its own enzyme. Although there are many reactions, the enzymes force all of them to take place over a very short amount of time. The third phosphate bond is a very high energy bond, and that is where the energy is stored. Take a quick look at this musical interlude about glycolysis.
Glycolysis is a series of reactions, enzymatic actions, energy transactions. Takes glucose, a molecule so sugary, breaks it down to NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes, organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis, come on sugar for the breakdown, for the breakdown.
Enzymes, in an AP chemistry student's terms, is a catalyst. It lowers the activation energy for a reaction, which makes the reaction occur much faster. A cell would not be able to survive without enzymes because most of the reactions in a cell take place very slowly without an enzyme. Enzymes ultimately do all the work in a cell. Each enzyme can have between 100 and 1000 amino acids bound together in a very specific order. There are hundreds of types of enzymes that float around a cell and wait to find the molecule it will recognize. The amount of a certain enzyme produced depends on its importance in the cell. Sometimes an enzyme's function is rendered useless when a substance known as an enzyme inhibitor binds to a receptor. That is why many poisons, such as hydrogen cyanide, can kill you. It disables a cell from functioning correctly. Here is a video to demonstrate the inhibiting process. The blue molecules are molecules that are supposed to be going into the enzyme. The red molecule is an inhibitor.
Could you lie